So, um, if you've ever really thought about the fundamental machinery of life, you pretty quickly run into proteins.
Oh, absolutely. They're the essential workers.
Right. They do everything from catalyzing reactions to providing structure, regulating DNA, even fighting off pathogens.
But there's this massive catch, a catch that has, it's stalled biology for decades.
A protein's function is dictated entirely by its final three-dimensional shape, its structure.
Exactly. And we live in this age of genomics, right? So knowing the sequence of amino acids, that one-dimensional blueprint, that part's easy. We know billions of them.
But figuring out the final folded 3D structure.
That's the bottleneck. Experimentally, using methods like X-ray crystallography or cryo-EM, determining just one unique structure it's a monumental effort. It can take months, sometimes years.
The numbers really do tell the whole story of this gap, don't they?
They do.
We have sequence data on billions of proteins, but the central database, the protein data bank, it only has about 100,000 unique solved structures.
Such a disparity, knowing the instruction manual versus knowing the final product.
So, okay, let's unpack this. The ultimate challenge, predicting that 3D structure from just the 1D sequence. It's known as the protein folding problem. And it's been an open research challenge for, what, more than 50 years?
Right. And that's exactly why we are diving deep into the source material from DeepMind today, specifically their paper, Highly Accurate Protein Structure Prediction with AlphaFold.
Which makes a huge claim.
A huge claim. It essentially says they've achieved atomic accuracy, predictions that are competitive with the structures that come from all that laborious lab work.
That is a tectonic shift, if it's true.
It absolutely is.
Yeah.
So our mission here is to really extract the core insights on how this neural network, AlphaFold, pulled this off, how it achieved this breakthrough performance, and what that means for the future of biology for you, the learner.
Okay, so to really grasp the significance of solving a 50-year-old problem, we have to look back. Look at the two traditional paths researchers took and why both of them ultimately hit a wall.
The first path was, you could call it the elegant but impossible ideal, the physical interaction program.
This involved trying to integrate all our knowledge of, like, molecular forces, thermodynamics, kinetics.
All of it into these massively complex simulations.
Intractable is the key word there, right? You're trying to model every single interaction between every atom at every moment.
Exactly. The computational difficulty was it was insurmountable, even for a pretty modest-sized protein.
And you're also trying to model the physics perfectly in the first place with all the chaotic effects of water and temperature.
The complexity just exploded.
So the field pivoted. It pivoted to the second path, the evolutionary program, or bioinformatics. This really capitalized on the explosion of genomic data.
Precisely. So instead of simulating physics, you look at evolutionary history.